This video is ECRs, adaptor proteins. So we're going to focus on adaptor proteins, which are just downstream from activated receptor tyrosine kinases. Definition for adaptor proteins is right here. Basically, the adaptor proteins facilitate activation of downstream intracellular signaling pathways through interaction domains. And they, on this figure here, are these little 1, 2, and 3 proteins. Those each are different adaptor proteins. One of the big things to focus on for this topic is domains. So I want to remind you about what domains are from previous parts of the course. So remember that domains, this is a part of a protein which has a specific function. So it's a discrete portion. And oftentimes these are conserved functions across different organisms throughout evolution. And that means that we can study them in a variety of different organisms, including model organisms, and then understand how they work within our own cells. Okay, let's talk a little bit more about the adaptor proteins. So a few things that you need to know about them is they have no enzymatic activity. And they basically work like a bridge, allowing two different proteins to be connected to one another and potentially to relay a activation signal from one protein to another when those two proteins could not actually recognize one another. These proteins are typically small, and then sometimes they're called scaffold proteins. This is typically when it's a larger protein, so a larger adaptor protein would be called a scaffold protein, but overall these terms of adaptor and scaffold are kind of loosely used, and so we'll basically use them interchangeably, but you should know both of the terms. The last thing is they are recruited when intracellular tyrosines are phosphorylated. And so, as you know, receptor tyrosine kinases are, through their activation, phosphorylated on specific residues like we see here. And then when they're phosphorylated, the tyrosines are, then these adaptor proteins, which might have been somewhere around here just kind of floating around in the cytoplasm, then they have a high affinity for the phosphorylated residues, whereas they did not have a high affinity for that before. One way we describe that is to say that the biochemical environment has changed, and so in particular we mean that these shapes are now able to stably associate with one another, and through this process of the adaptor proteins then lead to activation of other parts of signaling pathways. I also want to point out that these things fit together in incredibly and highly specific and predictable ways. So basically, if we talk about this adaptor protein 1, it can only recognize this particular tyrosine residue. And that would be based on the context of the other amino acids in this area. On the other hand, this adaptor protein 2 can only bind to this position, adaptor protein 3 only to this position. So even though it looks like they are all just kind of doing the same thing, they each have a highly specific role, and they cannot swap roles unless we mess around with some part of their protein structure. Okay, so now I want to talk about the interaction domain, and this is a very specific term, although it just sounds kind of vague, like it could be any domain. Interaction domain is a vocabulary term, and this is a part of an adaptor protein which allows it to bridge these different proteins in a complex. Two examples of interaction domains are SH2 domains and SH3 domains. Down here is the definition of interaction domain. So it's a specialized domain for recognizing and binding to specific structures within proteins. And again, an example of this is that interaction domain would be part of an adaptor protein. I want to emphasize again that they have no enzymatic function. They're just going to bring together different proteins. Okay, so let's take a look at these different domains. So here we have a protein structure. You can see the alpha helix, beta pleated sheet, and so this particular protein structure is the SH2 domain. I don't expect you to be able to recognize that, but just know that it has a discrete function and shape. The SH2 domain's role is to bind phosphorylated tyrosine residues. On the other hand, SH3 domain has a different shape, which you can see here. You should be able to recognize this as a protein, but you don't need to distinguish these two. The SH3 domain has a different shape and it also has a different role. Its role is to bind proline-rich regions of proteins. Proline is one of the amino acids. And what proline-rich refers to is that there might be a string of amino acids of which a high percentage of them are proline. And in this case, the SH3 domain could bind to that area. So basically, SH2 and SH3 domains can be part of an adaptor protein. They could each be a part of one adaptor protein, or there could be two adaptor proteins, one that had the SH2 domain and another that had the SH3 domain. I want to talk about the adaptor protein through an analogy of a plug adapter. And so over here, I don't know if you've ever been in a house or maybe a coffee shop in which you have a device that has a 3-prong power cord and you want to plug it into a receptacle, but that receptacle only has a 2-plug. It can only receive a 2-plug. And so there's these little plug adapters. That's what this thing is here. And so basically, it means that your device's power cord doesn't have to, and it can't, actually fit with the power source. So we have this little thing that can bridge. This doesn't actually do anything. It doesn't have any real capabilities on its own. So again, it's not in our analogy that this is not an enzyme, but it allows two things to work together that wouldn't have been able to work together or recognize each other otherwise. And again, with this example, so this is the adaptor protein. We would have a receptor tyrosine kinase receptor over here, and it would have some phosphorylated tyrosines. And so you could label based on what you know over here about what kind of domain that we would be talking about for here. And so that would be the SH2 domain. And on the other side of the adaptor protein, we have the SH3 domains. And so those would bind to proline-rich regions of an amino acid chain. And basically this protein that has the proline-rich domains has some way to be able to convey the activated message of the receptor tyrosine kinase through the adaptor protein onto whatever the next steps are. And again, as a reminder, here in my example, this whole thing is one adaptor protein that has SH2 domains as well as SH3 domains, but you could imagine a situation as well where there's two adaptor proteins, one of which has the SH2 domains and another one that has the SH3 domains. Here's an example of an adaptor protein that is conserved across many different organisms. It's called GRB2. Great name, everyone. And GRB2 stands for Growth Factor Receptor Bound Protein 2. Wow. You don't need to have the full name memorized, but it might help you to be able to connect up GRB2 with growth factor signaling through our receptor tyrosine kinases by knowing a little bit about that whole name. This figure is not from your textbook, but I like the amount of detail that it has. And so basically GRB2 is this shenanigans here. So GRB2 is this protein that has both blue and pink portions, and you can see that it's got labeled an SH2 domain, which is interacting with a phosphate, presumably on a tyrosine of this receptor protein. It also has two SH3 domains, and that is interacting with another protein, which is shown here in brown. The brown protein actually has a cool name, which is son of sevenless, this SOS. Or SOS. And it was named in fruit flies because fruit fly biologists actually will name things, will name their proteins names that people can remember and are kind of, I don't know, at least to me, interesting. But anyway, this is an example for what's going on here. So again, this is our adaptor protein. Which is GRB2. And it binds to another protein, which is typically called a scaffold protein. You could also call it another adaptor protein, SOS. And then through all of this binding, then they will activate the signaling pathway. Remember that the only way that the adaptor protein can be recruited to this enzyme receptor is when it's phosphorylated. So the only way this all happens is when the ligand is bound or the receptor is otherwise activated. And so with the information that there are SH3 domains, which bind SOS, then you would know that SOS has some proline-rich regions, presumably two proline-rich regions, which means that it can be recruited to the adaptor protein. I wanted to tell you a little bit more about why we're so interested in GRB2. So GRB2 is involved in development. It's involved in development across a variety of different organisms, including humans and various model organisms. And when people have made a loss of function GRB2 mutation in mice, it's actually lethal. That kind of situation makes it pretty hard to study the function of a given protein and try to understand what it's doing in development. And so scientists in that case were able to do a lot of their studies within fruit flies. So in fruit flies, GRB2 has a role in fly eye development. And so with a variety of genetic tricks, they're able to knock out GRB2 just within the eye, and because the eye is not a required part of development, then they could learn more about the pathway. And basically they used this study to determine the pathway and the mechanism, basically by sorting out the different components. So what are the components? What's the role for each? And what sequence that they're used in. And so this is one example about how it's really useful to have a variety of different model organisms and have this kind of buildup of different genetic tools. So I'm not going to go into exactly what those are, but just know that that is part of how we end up with this, that is the way we end up with this sort of example here, is to understand that GRB2 acts before son of sevenless. They would need to make double mutants and look at either the location or the effects of how the particular parts of the eye were made.